as Jake mentioned, Golden Helix, we've been around for 15 years. We offer software and services. I usually work in the services department, providing custom analysis to researchers all over. And more recently, since the start of the year, to be precise, I've taken a larger role also in interacting with some of our software customers, talking to people, helping out with people getting started in their analysis projects. And it's been a great learning experience for me to do that. I've been able to pick up on a lot of the issues that are important to people and see where a lot of people are struggling and making mistakes in their analysis. And we'll talk about some of those things a little bit as we go along. Now, just to finish off the introduction to our company, Snip and Variation Suite version 7 SDS is our main software package. We'll be looking at it a little bit today. It's a very powerful, very flexible and robust analysis platform for everything from data management to visualization to all kinds of statistics. We'll mostly be looking at just one area of functionality today, which is the DNA sequence analysis package. But we do, as Jake mentioned, have a lot of functionality in other areas and provide a lot of rich interactive visualizations, and we like to think it's a very empowering analysis experience. The other package we'll be looking at a little bit today is GenomeBrowse. GenomeBrowse is a completely free commercial-grade browser product that we provide. I won't say it's completely feature equivalent to IGV, but it has a lot of the same types of functionality that you would get with IGV, but with, we think, a much friendlier user experience, a more fluid interface, a lot more like Google Maps. That was kind of our target when we started, was to create the Google Earth of genome browsers. And we're still improving it, and we're really excited about it and hope that you find some use there. So before we get any further, I'm curious what the background is in our audience today. I mostly am coming from a statistics background, and over the years have been learning more and more about biology. I also know that in this field we find a lot of people with backgrounds in computer science and math and who knows what other sorts of training. So I'm curious of the people here, how many consider yourselves first and foremost a biologist or a wet lab researcher? So 3/4 of the audience. Okay. How many consider yourselves primarily a data analyst? So pretty much everyone else. Are there any clinicians in the room who are primarily in clinical practice? We have a handful. Okay, so as we go along, I'll try to point out some of the different themes that may be important to different sides of the aisle. And you'll also maybe see some of my own biases coming out as we talk in reference to the fact that I'm mostly coming from a quantitative background. So in next generation sequencing analyses, depending on who you talk to, you'll get different versions of what analysis means. And it mostly has to do with where that person sits. For those of you who are biologists, you'll probably think a lot more about what we're labeling as secondary analysis here. For those that are like me and more on the quantitative side, you'll probably think more about what we're calling tertiary here. But the basic difference is whether you're producing data or trying to make sense of the data. And in secondary analysis, that is what we label as all of the alignment or assembly if you're doing a de novo project, or then the variant calling that goes on after completing an alignment of all your short-read sequence data. And then in tertiary, the sense-making part, it's where we get into the phenotypic association and trying to find what is driving the disease or the other phenotype that we're studying. Now, there's a lot of different aspects to tertiary analysis, and tertiary is where we at Golden Helix primarily place ourselves. We don't get too involved in alignment and variant calling, except maybe to provide advice, and we do some of it in our services department. We don't really provide software there. But in tertiary, there's any number of different things people might be doing, whether it's in the clinical environment. We have some clinicians where you might be looking at a targeted gene panel, looking at variants or looking for variants that are known to drive disease. or you may be in more of a translational research project looking for the variants driving some rare Mendelian disease, maybe looking at nuclear families, different types of directions you may take there, or population studies. Usually this is getting into the complex disease area where there may be many different genes involved and you're dealing with issues of genetic heterogeneity, looking for rare variant burden within genes. Those are all things that we support in SDS and the kinds of things that I think about every day. And also in cancer analysis, looking for somatic variants, cancer tumor drivers. That's something that I personally have been trying to learn a lot more about lately, and something that we're hoping to provide better functionality for in the future. Now, as we talk about tertiary analysis and analysis in general, I went back in my archives and found a couple of old slides that I find to still be very applicable today, even though they're maybe a little out of date. So this is from 2 years ago. I was at a seminar sponsored by Illumina, and Jay Flatley, of course the Illumina CEO, got up and talked about the state of sequence analysis and pointed out that over the past decade, the greatest burden has been annotating the human genome, generating piles of reference data. And the burden now is shifting into analysis and bioinformatics, taking advantage of all of those catalogs of data that have been created. I agree with his assessment. I would say that that generation of data is still going on and still very important, and it's not something we need to let up on, but there's enough that's been done that we can start to do some really valuable research in additional genomes. Now, I can tell you this was actually somebody from Complete Genomics that I talked to about that same time a few years ago, who had worked with a number of scientists who were just getting started in sequence analysis, and his perspective was that most of them fell into two camps. That on one side of the aisle, he saw people who were coming into sequence analysis more from a GWAS background, primarily studying complex diseases, and they preferred to follow statistical algorithms to lead them to candidate susceptibility loci. While those who were coming from outside of GWAS, and he said it was really very distinct. You were one or the other. And from that background, they generally tended to prefer a data-driven, more bioinformatics approach, trying to directly identify causative variants. And at the time, he said that it's that second group that was being more successful, and I would say it's still the case. I know a lot of people looking at complex diseases are still dealing with power issues and trying to sort out the best approaches to take there. But at Golden Helix, as we've been designing our software tools, we've been doing everything we can to support both types of approaches and to integrate them as much as possible. And that brings us to this concept of bioinformatic filtering and annotation, or really it's what Jay Flatley was getting at, talking about using all these catalogs of information. And I would imagine that a lot of you here in the room today are already doing this kind of thing, but we have a wealth of data, huge databases coming from NHGRI and others that we can access to learn more about genetic variation. And some of it is strictly observational data, like variant frequencies from different population level projects. Other data is based on predictive algorithms coming out of bioinformatics community. And there's even simple things that maybe have been around for decades, like just simply knowing the mapped location of a particular gene. That is very valuable, and we may often take for granted that we know that. And so all of these different types of data can be brought in and used in an analysis project, and really at both the QC level and the analysis level. If you go back and look through different papers, you see people using bioinformatic filtering to, for example, remove common variation based on dbSNP or based on the 1,000 Genomes project if they are studying a rare disease and expecting a rare variant to be driving that disease. So that would be more of an analysis level of using it in that regard. In terms of a QC, you see people dropping variants that are in segmental duplications, known repeat regions, or near gaps in the human genome assembly. Those are areas that are known to be difficult to align short reads to. And so they decide to just ignore any variant calls that they have there. So that's more of a QC approach. And as we go along today, I'll show you some examples of how you can do both of those things in our software and using some other tools as well. So once we've annotated all of our variants, we can start to sort, filter, and prioritize to help us figure out what's going on and what's causing the disease we're looking at. And this slide, even though it says SVS in the first couple of bullets, I think applies to any analysis workflow. And study design is very important and something that we all need to think about as we're getting into sequencing. And in terms of how we work with SVS, there's a broad range of tools for manipulating data, for creating annotation files, and for filtering and exploring the data. But really, it's a matter of knowing what the question is that you want to ask and defining the series of steps that will get you to that answer. And so if you're looking for a rare dominant trait or a de novo mutation, or looking for either a recessive or compound heterozygous variant, in each of those cases you're going to take a slightly different approach. The workflow is going to be a little bit different to get there. And we just have to lay it all out from the beginning and go for it. And once you get that mindset, it's really, I want to say it's not difficult, but again, it maybe just depends on your background and what you're familiar with. And again, once we know what we're looking for, we can figure out which annotations we need and what sort of analysis tools we need to assemble into our pipeline. And so to start things off today, I want to walk through a case study for a disease that I don't want to say it has an easy answer, but it's one that's a shining example of what you can do with sequence analysis if everything works right. Of course, it doesn't always work out this well, but this is an example that was published about 2 years ago. And Jake already mentioned that the data for this project was shared with us by the senior author, Dr. Golson-Lyon. He's a friend of ours who allows us to use this for training and for demonstration purposes. And what we're going to look at today is a lethal X-linked disease that affected males in multiple generations of a family from Ogden, Utah. It was later called Ogden syndrome due to the geographic connection there. And we're going to look at sequence data from 5 members of this 3-generation family. So what we have is an affected male infant who died with this disease, together with his mother and grandmother, who in an X-linked scenario should be heterozygous carriers for the causal mutation. And we also have some negative controls here. We have a brother who did not have the disease, together with an uncle who also did not have it and therefore should not carry the variant we're looking for. So with that little bit of background, I'm going to hop out of PowerPoint here for a moment and switch gears. So I just opened the SVS analysis package. And what I'd like to do is start from the very beginning and just run through how a typical analysis workflow might go with this kind of a data set. We'll start by importing raw data from VCF files. We'll proceed to identify variants on the X chromosome that follow an X-linked pattern. We'll reduce that to the ones that are high-quality, non-synonymous variants, then annotate them and see if there are any clear and obvious candidates for causing the disease that come to the top. Does that sound like a logical workflow? Okay, good. So I'll begin by, as I mentioned, importing the VCF files that we need for this project. Now, I already mentioned we don't get too involved in alignment and variant calling. We generally expect that anyone using our software will already have VCF files produced by some variant caller. In this case, I have 5 VCF files, one for each of my 5 family members. And given that VCF is a really flexible format, there are a number of different fields that may be present in any given VCF file. I'm just going to pick a few of them. I'll get the genotypes, allelic depths, and just read depths, genotype qualities. With SVS, you just need to select which items you want to have available for analysis. If you need to go back and get others later, it's not too difficult to do. It's going to scan through those 5 VCFs and create a few spreadsheets of data that we can work with as we go along. This will just take a moment. Question? Yes? It can be single sample, multi-sample. As long as it adheres to either version 4 or 4.1 conventions for VCF, we can handle it. Any number of additional annotation fields, it's not a problem. We've spent a lot of time building a tool flexible enough to read any VCF. It's strange. I think VCF was originally intended to be machine readable, but it's mutated over time to be more human readable or Excel readable. And it makes it difficult to process sometimes with other functions. So you can see that since we have 5 different individuals that each had their own VCF, there's a lot of missing data. That's what all of these question marks are, where one individual has a variant call and nobody else had data present. And so what we're seeing if we open up this marker map area is one column for every genomic position where at least one individual has a variant call. Now, this particular data was collected using X chromosome-only exome capture, but there's still a lot of bleed over into other regions of the genome with the capture kit. You'll see that a lot. We probably have people here doing targeted sequencing analysis, but target capture is never totally perfect. So the first thing we'll do is just turn off everything but the X chromosome and say, from here out in this analysis, I only care about X. And you'll notice up here in the corner, it tells me a lot of columns just got turned off. And what that means, if you come over here into the autosomes, we see this gray text just indicates that that data has been filtered out and we're not going to use it anymore. You'll see as we go along that that idea of filtering based on either map position or based on external databases is something we'll use quite a bit. But now that I've reduced it just to the X chromosome, and I'm going to just rename this spreadsheet so I remember where we are here. I forgot to rename it on import, so it has that big long auto-generated name up here at the top based on the VCF file name. I should have adjusted that, but I didn't. Anyway, the next thing I want to do is figure out which of these variants are high quality. And back in the project navigator, when we first imported the data, we ended up with different spreadsheets for the read depth, for the quality score, and those spreadsheets have the same dimensions as my genotype data. But what I want to do is get back to that X-only spreadsheet. We have some tools that allow us to cross-reference between spreadsheets, drop out things based on different thresholds. So I'm going to do that. So in the QA menu, set genotypes to missing based on data in other spreadsheets. So I just need to specify I want to drop out low read depths and low quality scores. So it just takes me through a little wizard here where I can specify, okay, for read depth, what threshold do I want to use? I can either do a universal threshold or subset according to different zygosity states, use different thresholds. That's something we see a lot where they might require a higher threshold for heterozygotes. I'll keep it simple today. So we put read depth at 10, which is maybe pretty low, but it works well for this data. Genotype quality score, I'll set a minimum threshold at 20. So on the Fred scale, that's like 99% confidence, roughly. And so it tells me a bunch of my genotypes failed those thresholds, so nearly 4,000, and 2,000 columns of data are now entirely uninformative. They've been removed. But what I have now is just 3,000 variant positions that I need to sort through. Well, to begin with, we know it's X-linked, so let's set a filter here based on X-linked status. And there's a few ways to do it, but I'll use a really simple one here, which is, where did it go? Activate by sample genotypes. And so this is just a really simple little tool that lets me specify the exact zygosity state I'm looking for with each sample. So I'm looking for the affected male child to be homozygous for an alternate allele. It is X chromosome. Technically there's only one copy, but it's always coded as diploid here. We'll get the grandmother and mother to be carriers for the same alternate, while the two unaffected males are non-carriers, but they'll probably be coded as missing if they didn't have a variant column. And that's just because of the way the VCF files were processed. In other data sets, they might actually be coded as homozygous reference. So we can see only 67 variants past that filter, and I can subset to those 67, rename it one last time. Oops, X-linked. And I can look at these now and see that they all follow the pattern I'm looking for, that male affected child being homozygous with the carrier females being heterozygous. Another way to look at this is to throw it in the genome browser view. And this is our old genome browser. It's going to be replaced with a new and improved interface soon. But it is nice to just look at this even and see, looks like I have two chunks of the X chromosome that are segregating from the grandmother to the affected male child and not through the other lines. My background in genetic epidemiology is such that I like to look for things like this. If I saw things all across the X chromosome being passed through, I would really question it. I can zoom in around this area and pick a couple of these variants and see, you know, what else can we learn about them. Can't find my grab handle. There it is. So we can, whoops, zoom in here a little bit more. We can see that these variants are in an exon of this gene TCANC, and I could add additional annotations if I wanted to. So if I wanted to find out if these variants were common or rare or otherwise, I could maybe get variant frequencies from 1,000 genomes, variant frequencies from the NHLBI exome sequencing project, get functional data from database of non-synonymous functional predictions. And so working in this way, I can zoom in right around this particular variant, click on it, and see down here in my data console, it's a homozygous variant in the child. This one has never been reported in 1,000 genomes, but it's pretty common in the NHLBI exome project, so I'm not sure why it's missing from 1,000 genomes, but that happens a lot. Something we'll talk about. And this one also is non-synonymous, and I can come down to my prediction track and see SIFT calls it tolerated, and there's a number of other algorithms to look at. But what I really want to do is evaluate them all and, you know, create a table so I know more about all of these 67 variants. And I'll start here, and I know I'm going really fast. You don't need to worry about copying down all these steps. I'm just trying to demonstrate a workflow so we can all get our heads into the analysis game before we start hearing from some of our other guest speakers and different analysis projects. So with this classifier, it'll scan through my 67 X-linked variants. It goes pretty quick. Tells me two different reports. So I can see that most of these are, well, only 12 are coding, and then there's a number that are intronic, intergenic, other UTRs, and so on. Of those 12 coding variants, I can see, whoops, looks like there are 8 non-synonymous, 4 synonymous. I just want to look at the non-synonymous ones right now. So I'll activate the non-synonymous and pick those out of the parent spreadsheet. So from this X-linked sheet now, I see I just have 8 columns active. Those are my 8 non-synonymous X-linked candidates. So what can we learn about those? To begin, I want to find out if they are common or rare. So I have a tool here that will let me compare it to the NHLBI variant frequencies. So we'll just grab that track right down here. And I think I'm all set up to go, so we'll let this run. There's only 8 of them, so it should go quick. It tells me that 7 were found there. It gives me a report about those 7. And I can see on these frequencies, 46%, 45, very common, about 0.25% roughly. That's pretty rare, but not rare enough for this novel X-linked disease. But obviously there's one missing. Okay, so one of these up here has never been seen in NHLBI, and I can just eyeball it and know it's column 5 up here. It's this one that is not there. I could also cross-reference between the spreadsheets and highlight that column, but I already know which one it is. And maybe to just find out a little bit more about these 8, I'll run this tool to generate some functional predictions. And this will get me 5 different predictive algorithms that will look at all of these 8 variants. I'll merge this report together with the frequency data and keep unmatched rows. And then we'll do one more little merge here and create one great big table with everything we know about these 8 variants, at least everything that we've generated so far. And now I have a giant spreadsheet with 80 columns or 70 columns, something like that, but I can pick out this variant. I can see that 4 of my 5 algorithms predicted it to be damaging. See what the amino acid change is. Scroll across, see. So SIF calls it damaging, polyphen 2 probably damaging, mutation taster says disease-causing, mutation assessor calls it functional, which is their version of damaging. Fathom says it's tolerated, but Fathom's always kind of an oddball. We'll talk more about functional predictions later on. But then we see that that one is not in the NHLBI data, so we don't get a known canonical allele frequency there. We don't get any of this other great information that NHLBI gives us. And then we get over here to the end where we have our original classifications. We see it's in this gene, NAA10. Turns out that is the variant that was in this project eventually confirmed to cause the disease. And they did a lot of nice lab work to confirm that that was the causal variant. What I would like to do is maybe a little simpler than that, but at least, since we don't have time to go do any work with mice right now in the next couple hours, I will show you. Let's find the right project. So this is GenomeBrowse that we've already mentioned earlier. I can find the same set of samples, and let's search that gene, NAA10. Whoops, it can't find one of my tracks. That's okay. So I have up here at the top a track that represents all the variants called in that gene in the VCF file for the affected male child. It looks like there's only one variant in the child at all. We can zoom in here around it a little bit closer. And then we can kind of go down the way. So this first pile-up view we're looking at here. So this is all of the short reads. We can zoom right into the base pair level around that site. Let's set an anchor here so we can highlight where we're looking. So in the unaffected uncle, clearly no evidence of any kind of a SNP there. Similarly, the unaffected brother, we don't see anything going on. Here with the mother, looks like we have a nice heterozygous variant. That's exactly what we're hoping to see. I can click there and get information that out of 75 total reads for the mother, pretty even split between the reference allele of an A and the mismatch non-reference G. We go further down. We see also in the grandmother, nice heterozygous pattern, and in the affected male child, click here and see, whoops, where's my data? There it is, 56 reads. They all are for the G. Looks like a clean homozygous variant. If I zoom out of here a little bit, I don't see any evidence of poor alignments. I could add more annotations, find out if it's in a tricky region, around one of those repeat regions or one of those assembly gaps that we talked about, but it's not. This is one that looks good, and I trust it. So that was the 25-minute start-to-finish analysis that we all dream of being able to do, right? we all wish that everything was that straightforward of, starting out with about 100,000 variant sites, narrowing it down to 8 candidates, and having one really jump out as the obvious one. It doesn't always happen that way. Any questions about what you saw here? We'll talk more about the software later, but oh, so as I zoomed in here, so what we're seeing here, each of these blue or green bars is one of the reads that came off the Illumina machine. This one, I can't remember if it was done with 75 or 100 base pair reads, but for example, I could change the view on this and just stack it according to the paired ends, and then actually this one didn't have paired end sequencing, so that particular view won't work, but you get the idea. The blue ones are on the forward strand, green on the reverse strand, and that's what the coloring is. And anything that doesn't match the reference sequence is highlighted in a different color. So T's get highlighted blue, A's get highlighted red. The fact that this red is really pale indicates it's a low-quality base call. If we looked around and found a different SNP somewhere that was a high-quality base call, it'd be a really dark red. Here's a couple more that are kind of intermediate, but you can see the mismatches just get highlighted there. And that's what we're looking at. Okay. Yeah, in the back.
Can you comment on the nature of the filtering used for the quality of the base call?
So what I used on the quality was a GATK quality score. I set it at a threshold of 20, which, as I mentioned, is equivalent to about a 99% confidence. The read depth, I just kind of picked an arbitrary depth of 10 because I've looked at this data before and I know that there's a lot of noise below 10 if you compare the read depths to the quality scores, and if you look at especially outside of the X chromosome where there's where it was incidental coverage, a lot of those were down below 10, and you can kind of see that below 10 the calls just weren't very good here. So it was arbitrary. We can talk more about how you would determine what thresholds to use later, but one thing I like to do is just take the genome-wide coverage and make a histogram every variant, maybe even look at the coverage on homozygous variants in histogram versus heterozygous, and you can usually see pretty clearly what's good and what's bad. But we can talk more about that. Yeah?
So, where is it located for annotated database?
So everything that I'm doing today, right now, is on this laptop.
So if we purchase software, so all the databases are locally installed?
So if you were to get the software, let me point something out. If I come up here to this Add menu, what happens is we do have a cloud-based annotation server that has a bunch of annotations on it that you can either read from the cloud or you can download and have locally. I keep it local because it's more responsive and it shows up faster. You also, if you have SDS with it, you have the ability to create your own annotation files. Anything that you can put in a spreadsheet can be displayed. And we'll talk more about some of the nitty-gritty details as we go along. Yeah?
Are the functional scores from different algorithms generated on the fly?
Those were not generated on the fly. It's actually a curated database. So the question there was in regard to those functional prediction scores. Let me just close out of this menu. I think I even have a version. No, I don't see it right now. So let's add that in, and I'll show you how that was done. So I have this down at the bottom. Where did it go? DBNSFP functional prediction scores. So that exists as an annotation track that is curated to show available functional predictions at every possible non-synonymous variant according to ensemble. And so if we make that a little bigger, let's go back out. I can't remember where our variant went. We lost it. Okay, it's over here. So zoom right back in around this particular mutation. We can see here in the reference sequence that the reference allele is an A. What we actually have in this sample is a G. And if we come here and click and see what the A to G change is predicted as, this was all pre-curated, pre-calculated. It comes from database of non-synonymous functional predictions, which is maintained out of the University of Texas. Eric Boerwinkle's group, I think, does it. And so we just have all of those scores calculated. We're just picking it. Now, of course, there are situations where these scores might be out of date because it's a few months behind the official version. There are situations where there might be different transcripts of the gene that are in different reading frames where the prediction is going to be different. We don't account for all of that, but there's a lot that could happen. We'll take one more question, then we need to move on.
So for that, this is a cost. You said it's a cost.
This was determined to be causative.
Yeah, and originally you showed the table actually on all the scoring.
Mm-hmm.
You don't have any information on that, right?
Well,
I misheard or something.
Well, the reason there was no information for that one is that it was novel according to one database. There was one database that had never cataloged it before, and so there was no information showing up there.
But the secret polyphony.
Those are predictions, so it doesn't need to be observed in order to be predicted.
Right. So, but that doesn't give anything.
It did, yes. The symptom polyphen did give it, and we actually just clicked on the exact set of scores that it gave. And this is all abbreviated in this view, but you can see that the SIFT score there was 0, which in SIFT is damaging. Anything below 0.05 it considers damaging. And the polyphen 2 score being close to 1, that's what polyphen calls damaging. So they just have different scales. But we did have prediction information on it. But we'll talk after the break about predictions. You don't always want to trust them. They're a guideline. Okay. So let's take a minute here. You know, we've seen a good example, but there are a lot of things to be aware of as you are setting up an analysis project. And I'm just going to, oops, that's not what I wanted to do. I'm just going to close this project. Cancel. It's just, yeah, that is what I wanted to do. Cancel out of this and go back to our slides for a minute. Okay. So what I want to do is just spend the next, we're a little behind schedule, but the next 20, 25 minutes talking about some of the things to be aware of as you are setting up your project, and just to make sure that you know what you're getting into a little bit. I'm curious, of those of you here, how many have sequence data that they're analyzing today? About half, a little more than half. How many are expecting to get sequence data within the next 6 months to a year? Pretty much everyone else. Okay, so we're going to talk about just a few things you need to be aware of. And this, as Jake mentioned, is based on slides originally coming from Gabe Rudy, who is our VP of Product Development. As he said himself, he snores this stuff in his sleep. He gives this presentation much better than I do. This was a 3-hour short course that he gave at the XGen conference. I'm cutting it down to the 20-minute version, and hopefully we'll hit some of the highlights. So even though Golden Helix, we are really focused on the downstream tertiary level, what we need to talk about a little bit is secondary, and making sure that you do your secondary in a way that will make your tertiary as successful as possible. Now, depending on what your analysis project looks like, there may be some special considerations you need to take in the upstream phases, in the secondary analysis in particular. And really, when you think about all of the special cases that require a particular aligner or a particular variant caller to be successful, you start to get the idea that maybe there isn't just one standard upstream protocol. A lot of people have their one pipeline set up, and that's what they like, and so their friend says, oh, will you run my data through it? And they do, and then you realize, oh, wait a minute, I needed something different. So we'll touch on that just a little bit. One of the first major considerations you need to think about is your choice of reference sequence. Most of us are doing resequencing, where we're just sequencing data, aligning it to a reference genome. Most of the world right now is using GRCH37, which has been available since 2009. I would say it probably took about 2 years after that to really become standard. We're expecting a new genome later this year, and again, I expect it will take a while for people to really start using it, mostly because of all those annotations that we've already been talking about. It's going to take a while for the annotation community to catch up, and I think once we see most of those different things like the 1,000 genomes annotations and the functional prediction scores, all of those things we're talking about, once those start getting updated into version 38, I think everyone will catch on. But it's going to take a while. Be aware, though, that the major difference is, so far there have been about 200 patches that have been released for version 36, things that we know are wrong. Those are going to be replaced. There's going to be a few changes made where the allele in the reference sequence turns out to be extremely rare, and they're going to fix it to put in the more common allele. And the map positions are going to change a little bit from what I understand, just like when we went from 36 to 37 a few years ago. Now, what most of us are doing, not everybody, but most of us are really going to be primarily focused on single nucleotide variants, SNPs. It's the most common type of variation, and in many cases maybe the most easily identified. But be aware that there are a lot of different calling algorithms out there. GATK is kind of at the top of the heap and getting used the most, but it's not the only one available. And they don't always necessarily agree with each other. And I've seen, some people don't entirely appreciate this, and it is important to realize that depending on which caller you use, you will get different results. We can see in one particular sample, and this is published data, if you take the same alignment and run it through 5 different variant callers, there's only about 60% of the total number of variants that are called similarly on all 5 platforms. And if you extract from that just the variants that are novel in terms of not having been cataloged by dbSNP, it goes way down. Then the agreement becomes only about 11%. So if you're really interested in identifying a novel SNP that nobody's ever found before, that's where things start to get sketchy. And there could be other reasons for that. Might have to do with just low coverage in areas where SNPs aren't well cataloged, that kind of thing, but something to watch out for. One last note on variant callers. This is an example from Gabe, our product development VP. He had himself, his wife, and their child sequenced through the 23andMe pilot program for exomes, and found a number of interesting things as he reviewed the data. The example here, this is a situation where he and his wife were both homozygous for a non-reference allele, a different one. He was homozygous for a T, his wife for an A. If I remember right, the human reference there is a C. And what happened is their child, of course, was a heterozygote at that position, but the variant caller that was used called him to be homozygous for an A allele, which resulted in a Mendelian error being detected. And when he looked at it here, he said, wait a minute, this is just fine. Or, it should be heterozygous. And it turns out that that particular variant caller could not handle 2 non-reference alleles at any given site. And this kind of thing can happen. It's something to be aware of. So moving on, indels, that's another thing that many of us are interested in and learning more about. Normally, you have to think about indels as being smaller than cmds, cmds being bigger. But these are very interesting, potentially very functional in nature, but they're hard to call consistently. There's some difficulties there, especially one of the things that we have found challenging is inconsistencies in how an indel is placed around a homopolymer. So when you have either a short tandem repeat or just a repeat of the same base over and over, how would you call this second sequence if the first one is the reference? Is it an insertion at the beginning, at the end? Is it inserted somewhere in the middle? More and more there are standards that say it should be an insertion at the beginning, but we've found there's still inconsistencies in how that's applied. It's something to watch out for. One of our programmers actually wrote a rant about this on our blog last week, if you want to go look that up. But his complaint was that the standard nomenclature for annotating these is not sufficiently unambiguous. And if you try to put them in a database to be searchable and comparable with other sources, they don't always line up. So just be careful there if you're working with indels. It's the wrong direction. CMDs, won't spend much time on here, but you can detect CMDs in sequence data, especially if you have whole genome sequencing. It seems to work a lot better there. We get questions all the time from people who say, can I detect CMDs in my exome data? And there are methods out there for doing it. I'll say that some are more successful than others. I'm not sold that that is a solved problem yet. And the last type of variation people are looking at, structural variants, so large rearrangements, inversions, translocations, that kind of thing. Again, we're getting better at picking these out, especially with whole genome data. With long insert paired end sequencing is where it seems to work best. There's a lot of interesting examples in the cancer world with structural variation. The trick there is, well, not only here, but also in CMDs and with indels, so we don't have the same kind of rich annotation data available for analyzing these. And I would say we're still in the catalog generating phase with this stuff, as opposed to snips, where I think we're a lot more advanced. But it's an area that I find fascinating and wish that I knew more about. So we're going to switch back over to GenomeBrowse here for a minute and look at a few examples of how these types of variants will appear in high-quality data, and when you have a good example of any particular type of variant. So let's jump out of this and open up GenomeBrowse. And to begin with, I am going to switch to a different project where I have a whole genome BAM file. And some of these, well, actually they're all on a remote, not a remote drive, but an external hard drive that I've got plugged in by USB, and they aren't going to read and update really fast, so hopefully we can make this work well. So the first thing I want to look at, let me copy this position, is a structural variant. In fact, it's an inversion polymorphism. And we'll start by just looking at this histogram view here at the top. The thing that gives this away, if we zoom in right around here, we see that we have all of these that are in gray. That means that the reads in our pileup match with the reference sequence. Then out here we've got a bunch of colors indicating we have a lot of mismatches. And then similarly, come out here to the other end of the inversion, we see the same thing. But to really understand what's going on, and I can't see the little grab handle. This is one of those problems that I was afraid of might happen. Scroll down. Okay, there it is. If we zoom out and look at the paired end reads here, through most of the genome, we have blue, which is forward strand, which will then be connected by this gray bar representing the paired end insert, to green, which is the reverse read. But over here around this inversion, if you look at it, on the left, where we have reads spanning this one breakpoint, we have forward strand connecting through to a forward strand, while if it's spanning the right-hand breakpoint, we see reverse connecting through to reverse. And all of these mismatch alleles that we see are at the end of a read where it hits that breakpoint. And if we flipped it over, we would see that this particular sequence of going CC, TAG, CG, whatever, matches exactly what's on the other side of the breakpoint. It's just reversed. And so this is an example of what you would expect to see with a structural variant, in particular with a with this small inversion. With larger features, it might look a little bit different. One more thing I want to look at in this sample is copy this location, come up and paste it. I'll give you an idea. This is a copy number variant. If we look up here at the top across this coverage histogram, we see we've got pretty consistent, right around 40, 50x coverage. And then suddenly in one location it spikes up to almost double that. This is a CMD that was originally called, for this sample, using complete genomics. And again, I'm losing my mouse because I'm having to look around the corner of the screen here. We can see that what happened here is, well, it's kind of hard to see, and it's going to take a minute to update. But basically, we have an amplification event there, a duplication of that region. So let's switch gears and just look at a few SNPs in a family trio and get an idea of what they should look like. So go back over to my project with this trio data. And here, what I have, and again, I'm having a hard time seeing my scroll bar. There it is. So I have the pileup data for parent, and child here. And the first one I want to look at is just a simple SNP that follows the recessive pattern. So let's zoom in on this one. And we already looked at this a little bit with the Ogden syndrome data. This will take a minute for everything to update. But if we look closely around this one, we see this first parent has a nice heterozygote. Second parent, again, I'm struggling to see my scroll bar. There we are. Second parent also heterozygous, while the child homozygous. That one's clear and easy. And one that might be a little more interesting, though, is if we come down and grab here. This time we're going to see what a de novo SNP looks like. This particular locus, we're looking at the child. And the proportion might not be perfect, but we see 73 reads in a region that otherwise looks good for an allele, which, if we scroll up and look at the parents, is not present at all in the pileups for either parent. And it's always fun to find those because they are rare, especially in coding sequence. And this happens to be a non-synonymous variant that turns out isn't a particularly interesting one, but it's still a non-synonymous variant. The fact that it's not a perfect 50-50 proportion might indicate this is a somatic change that isn't in all of the tissues for this offspring. It's hard to know for sure without doing some more study there. So those are some good examples, and there's actually one more I want to show. And then we'll talk about some of the things that can go wrong. This last one is going to be a deletion. And this one is interesting to look at for a few reasons. So if we scroll up and look at the parents, we see both parents appear to be heterozygous for this deletion. We can zoom in on it. Deletions are just represented by this kind of purple bar. And again, I'm having scroll bar difficulties. When we come down and look at the child has an interesting situation. So as we look at it, we can see up here in the histogram view that not all of the reads are consistent with the deletion, but the child was called as homozygous. And we can look at this and see, for example, there's 3 reads here in the middle that aren't showing the deletion. I can zoom in on those. We find we're really close to the end of the read, and it's a repeat. So the deleted sequence is a TCT, and then it's followed by a TCT, TCT. Most aligners don't like to call indels unless there's no other choice. And here we have a deletion of a repeat sequence, but since this read wasn't long enough to span past the repeat, the aligner chose to just call it as not a deletion. So this is something to watch out for when you get into indels. Those repeats, we already said it's tricky with how to annotate them, but it's also tricky with how to align them and call them. And most alignment algorithms use this local de novo assembly or this local realignment around an indel. But even when you use that, you still run into tricks like this, where it's obviously a homozygous deletion, but it's difficult to show. So, Claire, I'm just going to take 5 more minutes. Okay. So let's just talk quickly about some of the things that can go wrong here, and then we'll look at maybe 1 or 2 more bad examples. I won't go into everything in this list, but it all comes from this great blog by Nick Lohman about the things that may go wrong in your secondary analysis or in your wet lab work. Everything from PCR duplication to bad alignments in the draft assembly that you're working with to align your data. Just take some time to read up on this if you are in that phase of your research. When you get your short read data, particularly if you're using an Illumina platform, you get this FASTQ format. You get it from other platforms too. One thing to be aware of, for example, if you have 100 base pair read lengths, the last several bases of a read, the quality always drops off. That's why with BWA, for example, it clips reads at the end of the read if the quality score starts to drop off. We've worked with people doing RNA-seq who say, I had 100 base pair reads. Why does everything look like it's been trimmed to 75? And we have to explain, well, if you look at this chart, you can see quite clearly those last several bases did not perform very well. Sometimes they are good. That's why we see the whiskers on this bar chart coming up pretty high, but you won't always get that. Aligners. I won't go into a lot of depth on aligners except to say that the basic issue there is we are making a trade-off. Really the gold standard for alignment was developed in the 1980s, the Smith-Waterman algorithm, but it's too slow to be applied on a genomic scale. And so we have a number of different algorithms that are available that usually are sacrificing a little bit of accuracy in order to have realistic speed and performance. And again, depending on whether you're working with DNA or RNA or have special needs with your DNA, there are different aligners you want to use. And if you want to know more about the best aligner to use, we can talk about it later. Indel realignment. I touched on this a moment ago when we looked at that indel where the deletion was, where the aligner chose not to call a deletion in some of those reads because it didn't quite span the repeat. A lot of aligners now and most analysis pipelines will use a local realignment around indels to try to clean that up. We'll see in a moment, though, that it doesn't always work, but most of the time it is really good. Variant callers, again, there's a lot of choices. GATK is definitely seems to be coming to the top of the pile. I'll warn you, a lot of people don't use it right. I won't go into the details, but it was designed for looking at 1,000 genome samples where you had low coverage and a lot of samples. A lot of people are trying to use it with high coverage and only a few samples. And you will occasionally get some bugs there. And you can learn about some of those bugs if you go to our blog. I'm just going to skip through some slides here. But Gabe, who is the one who produced these slides, wrote a whole blog about some of the issues to be aware of in GATK. Similarly, he wrote another blog that said, don't panic. There's a lot of good things going on. There's really a lot of positive and exciting developments, and you can have a successful sequencing analysis. So one of the most important things, though, is if you have a variant that your tertiary analysis spits out and says this is important, it's always good to just go back and confirm that there's nothing fishy about it. And just to give you an idea of some of the things to look for here, I'm just going to skip through a little further. I just want to show a few more things in the browser. So let's bring this back up. And we're still in this family trio. So we'll show some examples in this family. Let's start with this one. So this is something that was originally identified in a workflow where we were looking for interesting de novo snips. And we looked a minute ago at a de novo that looked like it was probably real. Now this one, though, if we give it a moment to come up, this is my external drive taking its time, we can see that, well, actually this isn't the, oh, I see what's going on. It was called two different ways. One of the callers actually said that the father also carried the allele. The other caller did not. But as we look at this, this is one where, if we scroll down to look at the child was called as a heterozygote, and you can see there's a pretty good match between, 50-50 split between the reference C and the alternate A. But you can also zoom in on it and see a lot of strand bias. There's no calls for that alternate allele in the forward strand. They're all down here in the reverse strand, and a lot of them are with low quality. That's why they're pale rather than being a nice dark red color. But then we can come up and look at, for example, this first parent. We see the same thing. Pretty low overall proportion, nothing in the forward strand, and on the reverse strand, generally called with low quality. So it's hard to know when you see something like this. Your caller might say, this looks like a de novo SNP. If you go back and look at it, there is some evidence that either the parent has it or that really nobody has it and it's just a sequencing artifact because of this low quality. Just something that's weird and hard to know exactly what's going on. Now, this next one, this will be the last one I show. I have a whole gallery of things that can go wrong, the rogues gallery, if you will. But what I want to show you is in a pair of cancer exomes, and unfortunately I'm zoomed in on the wrong place. This one can take quite a while to load up. But what I want you to see here, we can actually see it just with the histogram view. We don't really need to see the alignment. This was a location that appeared to be a couple of somatic mutations between a normal sample here at the top and a tumor sample at the bottom. And these samples had local indel realignment done using the exact same pipeline, run one after another, but the local alignments did not give the same result. What happens is that in the normal sample, it called a one base deletion at the beginning of this string of T's, and then a C to G SNP at the end. While the second aligner instead called the fourth T to be a SNP, changing T to G, and said that the C allele was being deleted. Now, if you construct the haplotypes and look at what's exactly going on here, both of these alignments give the identical sequence, they had, there's no somatic difference here. It is the same sequence both times, just two different ways of aligning it, even after using a local realigner that's supposed to correct all these problems. And it might take a while for these to come up. These are some gigantic BAM files that are sitting on an external drive that's really slow. So with that, I think I'll go ahead and rest my case. We'll save any questions about this portion until after the break. But we need to try to catch up with our schedule. Oh, you can see now how the alignments finally came up. So if we zoom in, we can tell that they're on the same haplotype. They're always on the same set of reads. Basically, up here, it's G, TTT, GT on that haplotype. If we come down and look at the other sample, so the tumor sample, and zoom in around it, we see that it's exactly the same. G, TTT, sorry, G, TTT, G, and then it skips over to the T. So two different ways to align the same little spot. It lights up and says, oh wow, there's this awesome somatic mutation, but when you dig into it, it's nothing of the sort.